the efficient assembly of supramolecular structures for minimal cell design and drug delivery remains challenging. Here we demonstrate efficient protocols that yield supramolecular structures based on amphiphilic elastin-like proteins.
The presented assembly protocols generate versatile supramolecular structures with adaptable physicochemical properties, such as vesicles, fibers, and coacervates. Protein membrane-based compartments demonstrate phase separation behavior and allow for the encapsulation of chemically diverse fluorescent cargo molecules.
For expression of F20E20M EGFP and F20E20M cherry, inoculate the main expression culture from the overnight pre-culture to an OD600 of 0.3. Incubate it in 400 milliliters of sterile LB medium supplemented with appropriate antibiotics at 37 degrees Celsius and 200 rpm. When the expression culture reaches OD600 of 0.5 to 0.8, add IPTG to a final concentration of 1 millimolar and reduce the incubation temperature to 20 degrees Celsius. For expression of amphiphilic ELP with the unnatural amino acid, inoculate the main expression culture from overnight E. coli preculture containing the appropriate plasmids to an OD of 0.3. Incubate it in 400 milliliters of sterile LB medium supplemented with kanamycin and chloramphenicol at 37 degrees Celsius and 200 rpm. To prepare the 100 millimolar unnatural amino acid stock, add 206.2 milligrams of unnatural amino acid to 8 milliliters of ultra-pure water. Raise the pH.